So today we're going to start off by continuing with renal balance 1. I looked through and I think that we don't actually need any of the other slides in order to continue our discussion of how water moves. And honestly, the main point of the kidney is to move water and reabsorb water. So I actually think it's not a bad place to start. So we're going to continue with that today. But I wanted to let you know how we're going to move through the rest of the unit. And I will renumber the slides in your folder. I just wanted to give you a heads up before I do that, because I don't know what your systems for organizing everything are. But after we go through renal balance, you're going to work on the anatomy of the urinary system. Actually, the way you're going to do that is on Friday, I'm going to be out. So you're going to work on an anatomy worksheet instead of lecture. I do recommend that you come into class and work on this together, and I'll have you send me either a screenshot or just like a photo of your notebook for full credit, just to make sure that we're on track with that. And that worksheet is going to cover some of this structure of the nephron stuff that we briefly traced out on Monday, as well as kind of recapping some of the anatomy that you're hopefully covering in lab during this unit as well. Okay. So just a note for that, any questions that show up on the test will be restricted to what's on the worksheet. So that's all you need to know, but I do have videos for the anatomy of the urinary system, so I'll post those for you as well in case any of you just prefer listening to me talk or want to kind of double check yourself. Okay. So you'll do that on Friday. We may have a little more renal balance 1 to finish up with on Monday, but after that we'll move into renal physiology. And we'll do renal balance 2, and then finally we'll end with acid-base homeostasis. So that should get you kind of back on track with what's going on with SI as well. I did let them know that we're starting in a weird place. So they are aware of that, but we should be back on track soon. Questions about the structure here? Yeah. Oh yeah, like anything that shows up on the exam about the anatomy will be something from that worksheet. Okay. Does that make sense? Yeah. Okay. And really the anatomy portion of this these lecture slides is really just to give you context for like when we're talking about the physiology part, like you don't know where you are in the body, it's kind of confusing. So really the anatomy portion is for context for the rest of the material. So to begin, since we're talking about water balance, we haven't yet talked about the anatomy. I just want to trace one more time the structure of the nephron, because we talk about specific locations within it. Okay. So we start with a kidney. That's something like this. Has an outer shell, that's our cortex. And then the inner part is structured in these renal pyramids. So you will be labeling these on your worksheet on Friday. So this is our medulla. Oops, I drew that triangle upside down. Okay, so cortex, pink, and medulla, the dark red. So if we zoom ourselves in, you're going to see that our nephron dives between these two spaces. So we have a cortex, and then we have the medulla part. That will actually continue much farther down. Okay. So the nephron starts out with a modified capillary called the glomerulus up in the cortex. And this is caught into a capsule, catching all sorts of fluid with all sorts of solutes in it, almost the entire contents of the plasma. So at this point, we start to call the fluid that moves into the nephron filtrate. So when we talk about filtrate, we're talking about this fluid going through. So as the nephron continues, we have a proximal tubule. So proximal always just means first, nearest to the beginning. And then from the proximal tubule, we go into our loop of Henle, or sometimes called the nephron loop. And this is where we dive into the medulla. So we have this long loop into the medulla. And we call these two sides of the loop the limbs. So we have our descending limb, so descending going down, descending limb, and we have our ascending limb. And we will be talking about what's going on with water in this location today. The nephron then continues. So because we had a proximal tubule, this means we also have a distal tubule. So it's kind of like a mirrored structure. And then we enter a collecting duct, and lots of other nephrons are going to pour into the same collecting duct. And that collecting duct and portions of that distal tubule also go through the medulla. So they continue down, and then ultimately the fluid goes to your bladder and out of your body. So the filtrate is flowing through the proximal tubule, then down through the loop of Henle that's traveling through the medulla. Then we end up back in the cortex briefly before diving back down into the collecting duct and out of here. So as we talk about water reabsorption, we're going to be talking about bulk reabsorption here in the proximal tubule. We're going to be talking about the role of water in setting up a gradient, the medullary osmotic gradient here through the loop of Henle. And that's less to do with water going back into your blood and more about how this whole thing is set up. Then we'll also talk about how hormones affect this area, the distal tubule and the collecting duct, to sort of modulate how much water gets reabsorbed. So talked about the fact that we want to be in water balance. So you want to adjust how much water your body is reabsorbing depending on how much water your cells are making through the process of metabolism and how much water you're eating and drinking. So the water reabsorption begins in the proximal tubule. It's going to be following solute reabsorption. And remember, when we say reabsorption, reabsorption means we're coming out of that filtrate, out of that fluid that's in the nephron, and back into the blood. And it's reabsorption because like you originally absorbed these materials somewhere in like your GI tract or something. So it's going back into the body like a second time, basically. So this is moving back into the blood, the water reabsorption. So our water reabsorption is going to follow our primary solute sodium, because as we have sodium move, wherever it moves to, that's increasing the solute concentration in that area. It's increasing the osmolarity. And osmolarity, we can think of higher osmolarity as like a stronger pull on water. So when we look at the proximal tubule, we're going to see that sodium is going to be actively transported across the basolateral membrane, and that's the one that's like touching capillaries, touching the blood. So sodium is going to be actively transported. We're going to use energy to move the sodium, but then once we move the sodium, the water can follow by osmosis, just follow that gradient that we've set up now and that higher osmolarity we've set up by transporting sodium. So we're going to take a closer look at that. But I'll explain this picture first, I guess. Okay, so just note, so the blue molecules that we're following here, or maybe they're purple, can't really tell the color. These are our water molecules. So we can see that first, water moved into our capsule, into our nephron, and it's moving along with a lot of other contents of the plasma. So it's now in the filtrate traveling along, and in the proximal tubule, this is the first place we see reabsorption back into the body. Okay, so what we're looking at here is a cell in the proximal tubule. So we're thinking about our nephron, we're right here at the beginning, basically, right? So right here at the beginning, that's our proximal tubule. We're looking at a cell in the wall. So our filtrate here is listed as tubular fluid. So our water and all sorts of other solutes, other contents of this fluid are moving along past the cell. The first thing we see happening is that we have, let's focus, we're focusing on sodium. We have sodium moving into the epithelial cells lining the tubule. And we have it being pumped using a sodium potassium pump into the peritubular fluid. So this is just a very small space between that epithelial cell and the capillary. And then we have the sodium moving back into the blood, so being reabsorbed into the plasma. So the first thing we're interested in is this sodium. Actually, let's stay on this side for a second. Okay. So a thing to note here, as I mentioned the other day, so whenever we see a red line in these diagrams, that's saying it's active transport. When we see green lines, that's saying it's passive transport. So really what's driving this process is our sodium potassium pump. And we've seen that process before, right? The role of the sodium potassium pump usually is to set up a gradient that makes other things happen, right? So what we have happening here is that our sodium potassium pump means that we're going to have a buildup of sodium where? Where are we going to have high sodium? It's super simple. So just follow the arrows, right? So our sodium yeah, our sodium is going towards the plasma. So the first place we're going to see the big buildup is in this peritubular fluid. So by contrast, we're going to have a low sodium concentration here in the epithelial cell. So that is why you see this green arrow for the sodium moving out of the tubular fluid, because it's actually responding to this gradient that our sodium potassium pump has set up. So it's moving towards this area of low sodium that was set up by the sodium potassium. And then on the other side into the plasma, it's green because we're moving away from this area of high sodium into the lower plasma. So that again is passive because it's down the sodium gradient. Okay. So this is like our first step here. So they've changed some of their symbols, but they're talking about the same thing here in this image. We're still looking at that epithelial cell. Just a reminder, apical membrane, apical, apex, that means it's like the top. So the top of the cell is facing the tubular fluid. Basolateral membrane, basal, like bottom, is the part facing the blood. So one thing we may have happening is just sodium going through its own transporter by passive transport, then getting actively transported into the peritubular fluid, passively transported back into the plasma. That's why it's just drawn X. That's the sort of like general format here. It may be co-transported along with another ion. In the previous slide, we saw an example of or counter transport with glucose. Any of those are valid ways of getting sodium into the cell. Don't worry about the specifics there. The point is just that the sodium is getting into the epithelial cell and then into the plasma, and we are using some energy to make that happen. So first thing, our solutes get reabsorbed, increasing osmolarity of the peritubular fluid in the plasma. So they're increasing osmolarity because osmolarity is a measure of like how much solute is there. So because we move sodium or potentially other ions into the peritubular fluid and into the plasma, we increase the osmolarity by increasing the solute concentration. Okay. So then the next pretty simple thing that happens is that because we now have this high concentration of solutes over here, right? So we have a bunch of sodium or other ions, right? We now have a gradient for water to move down. So essentially we have lots of water here, right? A high water concentration in the tubular fluid versus a lower water concentration in the plasma. Or we can think of it as water moving to water down those solutes that have moved into the plasma. So we just have passive transport, osmosis of water all the way across into the plasma. So it's just when we say it's following that solute concentration, what we mean is we had to move the sodium or the other ion to set up this gradient, and then the water could move because we set up the gradient of the ion. There is another solute that's moving along with water in this process, basically. So urea, which we'll mention very briefly again later, is a permeating solute. So permeating solute meaning it can cross the membrane here. So urea is also being reabsorbed passively during this process. So we're also removing urea from the tubular fluid here in the proximal tubule. So questions about that proximal tubule part first. So that's kind of our first step. This is our bulk reabsorption of water. This is where most of the water reabsorption is happening. About 70% of the water gets absorbed in this way. And there's not really any control on this process, right? We don't have any receptors receiving hormones here. All we have is our normal sodium potassium pumps doing their job here. So this is not a point at which we're going to control water reabsorption, but it is where we're going to have a lot of water reabsorption happening. So urea is what makes us call urine. So it will show up in the tubular fluid again. It's basically going to move back in at a certain point. Yeah. So, but at this point, it's permeating into the plasma. Mm-hmm. So now we're at a second spot in our nephron. So we're moving. We were just talking about the proximal tubule. So now we're going to talk about what water is doing in the loop of Henle. So this is as we're driving from the cortex into the medulla. Okay. So we're going to use osmolarity and changes to osmolarity, and changes to the movement of water in order to set up the medullary osmotic gradient. So medullary because we're talking about the medulla, osmotic because we're talking about the osmolarity, and gradient because we're going to see that we have increasing osmolarity as we move deeper and deeper into the medulla, which is basically like deeper and deeper into the kidney. So the osmolarity of the interstitial fluid of the medulla, so the fluid around the cells here in this region, is going to change depending on how deep down you are. We have lower osmolarity up here by the cortex. And we have higher and higher and higher osmolarity the deeper and deeper we go, down to about 1400. So the greatest osmolarity is going to be at the deepest locations of the medulla. This gradient is going to be important for water reabsorption and water movement out of this tubule. We're going to zoom in for a second and note a couple of things about this state. And should note that in the slides, we're going to go through one of these sort of like thought experiments your book likes to do. Your book likes to explain to you how you start out from a cell. It's just like your basic normal cell with no differences across the membrane to how we set up a steady state that exists in the body. We kind of talked about this when we talked about electrochemical gradients in neurons. So we're going to talk about how this gradient gets set up. But what's probably most important for you to know for the future is the actual steady state that you would see in an actual human right now. So that's what we're looking at here. And we'll go through the slot experiment and see how we set this up, basically. So what we can note here is that in our cortex, we have this 300 milliosmoles of osmolarity, right? And we mentioned on Monday that this is the normal osmolarity of most body fluids. So we're going to see that that's also actually the osmolarity of the tubular fluid. So in the cortex, we're at this normal state. As we dive deeper into the medulla, we are increasing in osmolarity. So basically, we have more and more solute buildup, exerting a stronger and stronger pull on water. And the descending limb of the nephron is responding to this gradient with water moving out. Okay, so because we're increasing the osmolarity as we go deeper into the medulla, the descending limb is permeable to water, so water moves into the medulla. In the ascending limb, what we'll find is that the ascending limb is actually impermeable to water. So water cannot cross the membrane of the thick part of the ascending limb specifically. So these membranes basically can't do osmosis. They don't let water across. So we won't see water movement here in the ascending limb. But what we do see is active transport of solutes. So that active transport of solutes is how we set up this medullary osmotic gradient. So by actively moving all these solutes into the medulla, we increase its osmolarity, which is what lets us pull on water with the descending limb. And to kind of contextualize this, because it can seem a little sort of flipped if you're just following the structure of fluid moving through the nephron, that we move the water and then the solutes. So it's important to realize that in this region of the medulla, there are many, many nephrons, and they're all next to each other, and the space is pretty small. So there might be another nephron over here putting a bunch of solutes into this region. So don't think about it as divided in half. This is one of those places where, like we talk in phys lab about the fact that we have to remember tissues are three-dimensional and any picture we see is just a slice through it. That's kind of what's going on here. Okay. So the osmotic gradient is established by something we call the countercurrent multiplier. So that's like a key term. Probably should have made it red. So countercurrent multiplier. What the countercurrent multiplier is referring to is the fact that we basically have this movement of fluid in opposite directions through the loop, right? So in the descending limb, our fluid is moving down, and then in the ascending limb, our fluid is moving up. And we have water moving across the descending limb, solutes moving across the ascending limb, but they have this sort of opposite direction. And the action of these two things in opposite direction helps us establish this osmotic gradient. So ascending limb can't move water, but it actively transports solutes, mainly sodium and chloride ions, but also some potassium ions. The descending limb does not transport solutes, but what it can do is move water. So it's permeable to water. Only the descending limb is permeable to water when we're talking about the loop. So our overall result is that our fluid entering the loop of Henle from our proximal tubules is at our sort of base state of 300 milliosmoles. But as we move into the medulla, which has higher osmolarity, the fluid is increasing in osmolarity because as you take water out of that fluid, you're leaving behind a bunch of solutes, right? If we're only moving water out of the descending limb, that's essentially concentrating the sodium ions that are left in the fluid, increasing that osmolarity in the descending limb. And this keeps happening until we get to the point in the ascending limb where we start pumping out those solutes. At which point now, because we're removing all those solutes that got left behind, that got concentrated in the filtrate, we start to see that osmolarity go back down to the point that actually, by the time we get back up into the cortex and continue moving through, we're going to see that we are going to be at an even lower osmolarity than we started at. So we're going to see that in the cortex and the proximal tubule, cortex and the filtrate have the same osmolarity. But in the distal tubule, as we move here, cortex is still going to be at 300, but we're going to have an even lower osmolarity in the filtrate at this point after it's been through the loop of Henle. Yeah.
That they're actively being moved in the thick part of the ascending limb, is that correct?
The solutes? Yes.
Are those proteins, or is it very important to understand what type of active transport it is?
Oh, yeah, they're just pumps. So they're proteins embedded in the membrane. Yeah. Yep. Okay. Yeah, so when we're starting out, the osmolarity is the same as the interstitial fluid in the cortex. This is. Then as we move through the descending limb, the osmolarity is increasing. And then as we move through the ascending limb, it's decreasing. And we're going to see that there's also a relationship between the osmolarity of the medulla and the osmolarity of this fluid, where the osmolarity of the medulla through the descending limb always has to be greater so that we can still have movement of water. So the easiest place to note that is we noted on the previous diagram that at the deepest points of the medulla, we were at 1400 milliosmoles. But you can see here at the deepest point of this loop, we're at 1200. This means that there is still some gradient, some force for water to move out here, right? So we're always going to see that we're at a slightly higher osmolarity in the medulla compared to the fluid in the descending limb. So the result overall of this countercurrent multiplier is that the fluid in the distal tubule, which is one of the places where we're going to be able to control reabsorption of water, we'll see the fluid in that distal tubule has quite low osmolarity. It's 100 milliosmoles. So it has a lower osmolarity than the surrounding cortex, which means that if water is able to cross across the walls of that distal tubule, it'll move out of the distal tubule and into the cortex surrounding it. The same thing is going to actually happen in the collecting duct, which travels back down through the medulla again. And we need to remember that this gradient was set up here in the medullary interstitial fluid, where we have increasing osmolarity as we go from the cortex, so that's the shell of the kidney, to the renal pelvis, which is just like, the kidney is shaped like a kidney bean, right? So the renal pelvis is just that sort of little divot in the center, a little bend. So you'll see that. So now we're going to walk through how this got set up. So this is all showing us how we start out with a situation where there's no gradient anywhere, and how this process eventually establishes the osmotic gradient. And then this final image is showing us that system in steady state. So we're going to go through all of this, but if you get lost, focus in on this final step because that's what we actually see in patients, basically. So we're going to start out with a situation where everything is just at our baseline 300 milliosmoles. So we can see here, we have our cortex at 300 milliosmoles. All through the medulla, we're at 300 milliosmoles. Our fluid is at 300 milliosmoles. So everything here is in equilibrium at the beginning of our thought experiment. So at this point, is there any force for water to move anywhere, basically? No, right? There's no gradient. No water is going to move here. So the first thing that needs to happen to set up our gradient is that we need to pump ions. Right, so the first thing that's going to happen is this active transport. So active using energy, active transport of those ions from the tubular fluid, from this filtrate into the medulla. So we can then see the results here. So we're going to note the differences first that we've created by doing this. So one thing we can see has happened. So before, if we're looking at the ascending limb, we're at 300 and the surrounding medulla was at 300. But we've moved sodium chloride and potassium out of the fluid and into the medulla. So we can see the osmolarity of the fluid went down. Right, so we're at 300 before, now we're at 200. That's because we moved those salts out of the fluid and into the medulla. So we can also see a change in the osmolarity of the medulla in this region. So we're now at 400 instead of 300 because that's the location that we moved these ions to. So we increased the concentration of solutes in the medulla, which increased its osmolarity. Now we do have a pull on water, right? So we see that the tubular fluid has an osmolarity of 300 milliosmoles, which is less than the medulla, and it's 400 milliosmoles. So now water is able to move by osmosis towards that greater osmolarity. And this is happening only in the descending limb, so only in this limb that's going down, because that's the only one that has membranes that are permeable to water. Usually when we talk about cells in basic biology, right, we just kind of like assume that they are permeable to water, but that's not actually always true. So water can't actually pass every single membrane. So it can pass the descending limb, but not the ascending limb. So our next image shows us what happens because of that movement of water, basically. So we've now reached equilibrium between the descending limb and the medulla. So now we're at 400. We just left it here in the medulla. Medulla is big, has lots of stuff going through it. And the fluid in the descending limb has reached equilibrium, so water stopped moving. Our ascending limb, nothing happened, so we're still at 200. And all of this is only happening in the medulla, so note that in the cortex, nothing is different between these images. So we're just staying at 300 in the cortex. So now what we're adding on to this is that in these first three images, we were pretending that that fluid was just kind of standing still in the nephron, so it's standing still in this loop of Henle. But now we're going to add on the fact that fluid is actually being pushed through this loop. So what we see happening is kind of like when we talked about breathing in fresh air into the alveoli and we saw mixing there. So what we're seeing is pushing this fluid that's at 300 milliosmoles deeper into the medulla, right? So now we can see it's at 300 down to about here. And then we can see our region of 400 beginning. And we can see before the 400 was only until the bottom of the loop. But that whole kind of stack of fluid has been moved up. So now it reaches a little farther up towards the ascending limb before we get to the point where we have the active transport of solutes. Okay. So now we continue to have osmosis happening. So we had, just a reminder, we were at like 400, I believe, in the medulla in our previous image, right? But we continued to have water move into the medulla. So now we are lowering the osmolarity of these more sort of surface levels of the medulla. But as we get deeper into the medulla, we'll see that we always need a gradient for water to move. And there's less of a force for water to move as we grow deeper into the medulla. So more water is able to move across and water down this kind of more superficial portion of the medulla. But at the deeper limits, we have less water able to move. And also we need basically more of a buildup of solutes in order for any water to move. So this is starting to kind of set up that gradient because we have more water moving higher up in the medulla, a little bit less water moving deeper into the medulla. So now we start to see that gradient. Yeah. Okay. So we can see that that gradient is also existing within the tubules. And what this image is showing us is just that after this water movement happened, again, we came to equilibrium, but now we just came to equilibrium with the surrounding areas of the medulla. So this can tell you that this fluid is not moving all that fast in our imagination here. And what I would also like us to note is that as we're moving back up into the cortex in the ascending limb, we're now also kind of pushing this low osmolarity fluid up into the cortex, right? So we can see this starting to drop here because we're pushing through fluid that we've removed many solutes from. So this continues to happen over and over again until we reach our steady state, which is the one we want to really be familiar with, which is where our cortex and our proximal tubular fluid is at 300. But we have an increasing osmotic gradient established as we move down the medulla. And the same thing we see in the descending limb. But as we move into the ascending limb, we start to see the tubular fluid with much lower osmolarity than surrounding regions of the medulla because we're actively pumping those solutes. And because this fluid gets pushed into the distal tubules in the cortex, we then see a difference between the osmolarity of the fluid in the distal tubule versus the interstitial fluid in the cortex. Questions about this process? Yep.
So are we gonna want all of those numbers memorized, or just the fact that what goes up and which goes down?
I would remember what goes up, what goes down. I would remember 300, 1400, and 100. Okay. So only numbers from the steady state and only kind of maximums and minimums. So that's how we reached our site. Mhm.
Sorry.
Yep.
Is it just moving in, or are they also like moving outside the loop? Because in the pictures they're moving in, but some of the pictures we looked at earlier, they were pulling them out.
Sorry, what's the is what move? What are you talking about moving?
The solutes in the water.
Okay, yeah. So the water and the solutes are moving into the medulla here. Yeah. At this point, they're not moving into the tubular fluid. Is that what you're asking?
I'm sorry, like on the inside of the loop versus the outside of the loop. Is there also like a active process that moves water to the outside of the loop? Because in these pictures, they're just moving towards the inside of the loop.
What do you mean by the outside of the loop? Sorry.
No, you're fine. Just in like the earlier pictures, the arrows are pointing outside of the loop, like towards the.
Let me see. Like this one?
Yeah. So it's going like inside the loop and then outside of the loop.
So what's happening overall is that we're moving out of the nephron, so out of the loop of Henle.
Okay.
So that's both the water and the solutes, right? All of those are just moving out. What we are doing is moving into the capillary. Is that what you're asking?
Yeah.
Okay.
Sorry.
Yeah. We're gonna get to that next. That's fine.
Okay.
Mhm. So one more note before we actually do start talking about those capillaries is that role of urea again. So urea is generated by the liver, that's where it comes from, and it's one of the ways your body gets rid of nitrogen. It's very water soluble. Now, in order to move urea, you do need urea transporters. So it was a permeable solute in that proximal tubule, but that's because the transporter does exist in the proximal tubule. Don't worry about the difference between UTA, UTB, and UTC. Just think about those as urea transporters. So here we can see that urea was able to move in the proximal tubule because of the presence of those transporters. But we're also going to see that those transporters are present in the collecting duct and in capillaries next to the loop of Henle, so peritubular capillaries going through the medulla. So this is also adding to the gradient that allows water to move, basically. So this is partially adding into the osmolarity of the medulla. So it's not just sodium chloride and potassium. It is also that we built up urea here in the medulla because we suddenly are able to move urea again as we go deep into the medulla with the collecting duct. So I want to take a second to trace the blood flow. Okay. So far, the only place we've named our blood flow is the glomerulus, right? So this modified capillary, we had most of the contents of the plasma turning into filtrate in our nephron. I'm going to overall trace the blood flow, how it got there, and then how it's leaving. So we have an arteriole that went into the glomerulus called the afferent arteriole. Like we talked about afferent nerves, A always means 2 here. Then we had the glomerulus, then we had the efferent arteriole. And we'll talk about those 3 structures a lot when we get to the renal physiology portion. But efferent means away. Then we'll note that this blood flow does continue. So we continue to have capillaries moving. So we have some branches that swirl around our tubules. Both the proximal tubules and the distal tubules are kind of next to each other because of this loop situation. Right? So we have peritubular capillaries surrounding them. And then we also have these structures called the vasa recta that are next to our loop of Henle. Okay. So note that they are first passing down the ascending limb, basically. So again, we have this sort of opposite current situation where we have fluid going up the ascending limb, but right next to it, we have blood flow going down the vasa recta, so down the blood that's passing next to it. And then fluid goes up on the other side where we have fluid in the descending limb going down. So this is another sort of component of this counter current multiplier. And just because I think you are talking about GI stuff in lab right now, do note that vasa recta, all that means is like straight vessels. So these vasa recta are not the same as the ones you're going to see in lab. It's just kind of a general term. Anytime you see like a straight blood vessel, they may call it that. So this arrangement of the vasa recta, these capillaries next to the loop of Henle, prevents the gradient from basically getting dissipated, getting erased by everything coming to equilibrium. The fact that we have this movement of blood flow in the opposite direction next to it. I'd also like to point out about this figure because I found this confusing when I first saw it. Despite the fact that we've been looking at pictures of the loop of Henle before, they didn't draw the loop of Henle on here. They are only drawing the blood flow on this image, which is why all the numbers are different and why the arrows look different here, because this is actually showing you the vasa recta, the blood flowing through the medulla in this region. So we see somewhat similar things happening. So in the cortex, we have our plasma, blood at 300 milliosmoles in the vasa recta. As the vasa recta descend into the medulla, we start to see them respond to that gradient. So we start to see water moving out. But now we are talking about blood vessels, so they have different properties. So in this descending part of the vasa recta, we also see solutes moving in. So that's part of why we don't see the exact same numbers here, because we are allowing water to move out and ions to move in. Okay. So water is leaving by osmosis, solutes are entering by diffusion. And then in the ascending limb, this is another difference from the loop of Henle. In the ascending limb, water is able to move into the plasma, right? So water can still cross. It can cross both of these limbs of the vasa recta, and solutes are moving out. So we have both solutes and water crossing in the vasa recta in both the descending and ascending parts. Another thing to note here is that because water is moving across both these limbs, this isn't going to change the gradient, the medullary osmotic gradient much, because the water that is leaving the descending limb of the vasa recta goes across and then moves back into the ascending limb of the vasa recta, so up that capillary again. So we're in balance here basically, right? We're adding water into the medulla, but we're also removing it on the other side of the vasa recta. This is a place where we do see urea able to move, or sorry, urea has moved into the medulla, but we have higher osmolarity in the higher osmolarity here in the blood because of the lack of the urea transporters. Okay, so we'll stop here and we'll pick up with the distal tubule and collecting ducts on Monday. So I'll post our worksheet for anatomy stuff for Friday. Otherwise, have a great weekend. And let me know if you have any questions as we move forward.